Myself, Dr. Humaira. I am from J.A.T Arts, Science and Commerce College for Women, Malegaon, Nashik. My topic is steps and precautions of section cutting. For formation of a permanent slide, one main step is section cutting. So sectioning is a process in which tissues are cut into uniformly thin slices or sections with the help of a machine to facilitate the studies under the microscope. The machine or instrument used for cutting sections is known as microtome. So, for section cutting, some steps are useful to follow it for better section, and its precautions are also there, which is helpful for better section cutting. So, first of all, the paraffin embedding technique is an ideal and universally used technique for preparation of permanent slides. The steps involved in this technique is collection and killing of material. The healthy material should be selected. The animal can be killed either by anesthetics or narcotics. After killing the animal, it is dissected and required tissues is cut into smaller pieces and preserved in fixatives for microscope slides. The next step is fixation. The preservation of the cells and tissues for cytological studies is called fixation. It prevents post mortem changes like bacterial decay and autolysis. In fixation, the tissue material is treated with the help of a chemical agent for preserving the morphological organizations, chemical composition, and preparing it for further process. For that purpose, fixation process is used. The next step is washing. After fixation, the traces of fixatives should be removed by washing the tissue in water. Washing is carried out in running water, keeping the tissue in beaker covered with cloth. Washing is done to remove oxidizing agents to prevent reduction when coming in contact with alcohol. Alcoholic fixative should be washed with alcohol. After washing, the dehydration or removal of the water is main step. It is necessary because most of the media in which tissue will be mounted are not miscible with water. Dehydrants cause considerable shrinkage of tissue. Improper dehydration causes pulling away the cell contents from the cell wall and also leads to shrinkage and brittleness in paraffin. Ethyl alcohol is widely used dehydrant, but isopropyl alcohol, acetone, dioxane, methyl alcohol also used as a dehydrant. So dehydration is necessary. After dehydrating, clearing or dealcoholization is the next step in which removal of alcohol from tissue blocks may be immersing. As alcohol is not miscible with paraffin, it must be replaced with a solvent miscible with alcohol and paraffin. The clearing agent makes the tissue transparent, increase the refractive index of the tissue. The presence of water or alcohol in the tissue may lead to crumbling of block during sectioning. The clearing agent should be changed in order to remove all traces of dehydrating agent from the tissue. Hence, the clearing and dealcoholization step is used. After that, the next step is Impregnation. Impregnation consists of replacing dealcoholizing or clearing agent with paraffin. The tissue is transferred into dealcoholizing agents saturated with paraffin and left overnight at room temperature called cold impregnation. The paraffin enters the cells, then hot impregnation is carried out, essential to get rid of all traces of clearing agent. The next step after impregnation is embedding. It includes pouring out the melted wax and tissue in suitable receptacles and arranging the tissue block in proper position and then cooling. Paraffin wax is most commonly used embedding material. After embedding, section cutting is the necessary. For section cutting, the block must be trimmed by keeping appropriate shape and size and fixed to the microtome page. The sections in the form of ribbon should adhere to the slide. Apply adhering medium, that is Mayer's albumin, on killing slide. Take the pieces of ribbon on slide. Put a drop of water and heat gently the slide for making the tissue straight. Then keep the slide slanting for drying. This whole process is called affixation. So affixation is necessary for the attaching of the slides on the or ribbon on the slide. Before staining, dewaxing, that means removal of wax by xylene and hydration, in which slides are processed in a series of alcohol in descending order. The next step after affixation is staining. It consists of staining proper, de-staining and washing. Different stains are used for staining. After staining, dehydration and counter staining is necessary. It is carried out by passing section through ascending order alcoholic series, and then eosine is commonly used as counter stain. After staining, the clearing of the slide is necessary, which is performed by the help of xylene, and place it on blotting paper. A drop of mountain in the form of the Canada balsam or DPX is taken and place the cover slip without air bubbles. Then keep the slide for drying. In such a way, the precautions and steps for section cutting is completed. After that, uh, we are ready to see the sections in the microscope.